we started off talking about Hodgkin's disease, and we were talking a little bit about the differences in the microenvironment. But I think it's important to spend a little more time on the trials in Hodgkin's disease. Recently we've seen the FDA approval of nivolumab for refractory Hodgkin's disease. Can you talk about that?
Sure. So as I mentioned, there was a signal early on in the phase 1 across the board in non-Hodgkin lymphoma, but at the same time in Hodgkin it clearly appeared on a small number of patients, 25 patients that were published in the New England Journal of Medicine, where the response rate was 87%. And patients were treated until achieved a CR or had toxicity or stopped the treatment. They were not treated indefinitely, and some of these patients have remained in remission up to 2 years. And up close to 20% of them were in CR. So this was really remarkable in a heavily pretreated population that is not necessarily a candidate for allogenic transplant or has any other option. As I mentioned, most of them had failed prior brentuximab. So this is clearly something that was such a strong signal. And we see more and more in the field of the pipeline of oncology and hematological malignancies, we get drug approved early on based on phase 2 data because they're so strong.
Did that surprise you? It surprised me. I mean, with that beat up patient population, I would not have thought that an immunotherapy has so much success. We think of all the therapy these patients get. It's directed at the lymphoid space. It's very lymphoid depleting, yet it's still effective. But
it's interesting what's happening in Hodgkin lymphoma. You're right, but when you think about it, patients with Hodgkin lymphoma can be severely immunosuppressed, but their microenvironment is very peculiar. They have very few tumor cells, rich tumor cells, particularly at the beginning. Over time, they have more. But you have this whole infiltrate that varies from mostly a T-cell infiltrate or a mixed B and T cells that are essentially there to promote the tumor cells. And so having a way to try to navigate this infiltrate and direct them against the rich tumor cells, it was not necessarily predictable, but very impressive and makes sense a posteriori.
And I think, let me just add that I think with respect to knowledge gaps, I think it's very exciting to see that, we saw these responses in melanoma and lung cancer, and then we're really starting to see the growth of this correlative scientific, understanding of what is the composition and what predictors happen. In Hodgkin's disease and lymphomas in particular, we have the ability to, in that initial biopsy lymph node, to really characterize how that tumor microenvironment is assessed on a cellular level will predict responses. And we haven't really seen, those detailed analyses coming out, but I think that when that happens, that's inevitable with all the exciting clinical trial activity. It'll really help us to guide, predictors of response.
Well, it's a very exciting field in Hodgkin lymphoma that hasn't seen a new drug for decades, and all of a sudden we have brentuximab obviously recently that is an anti-CD30 antibody drug conjugate with impressive activity, and it is now moved into the frontline combination. But having checkpoint inhibitors, I think the future is really exciting because we're going to be able, for example, we actually have an ongoing trial that is nivo plus brentuximab in patients who are not candidate for standard chemotherapy, elderly patients. And I think we really are going to see an interesting shift on how we treat those patients. And we know Hodgkin typically does well with ABVD, but there's a fraction of 10-15% of patients who are very refractory. So this is great to have new tools.
And I think it's important that we take a step back. Hodgkin's disease, it makes sense based upon the preclinical data that patients would respond. Again, based upon that 9p24.1 amplification, which drives PD-ligand 1 and PD-ligand 2. That amplification also drives JAK2 expression, which also increases PD-ligand 1 expression. So that it makes sense for Hodgkin's, and that rationale can't necessarily be applied to the other hematologic malignancies. And as we've seen with single-agent checkpoint inhibitors, the response rates aren't as good for follicular lymphoma and diffuse large B-cell lymphoma, or I'd also point out multiple myeloma. As a single agent, PD-1 antibodies against PD-1, there's really no response.